Good morning to all of you. First of all, I would like to give thanks to my Lord Jesus Christ for having this beautiful and blessed moment here. And respected dignitaries, chief guest of the occasion, Honorable Minister, Ren N.B. Dahalju, Science and Technology Minister, Honorable Minister, Ren Laydong Lepcha, Honorable Minister, Ren Laydong Lepcha, Patron Ren Kunganima Lepcha, Ren Sitin Rongkup, Ren Sitin Sandyang Rongkup, ITS dignitaries and all my loving my brothers and sisters. So, ngulin nahan go semnong lamdo toxic future, surum gun abi sorongsa Tendong knowledge series team gun team room. Kusum abi CM no abi lat na kusum ari tezet ka sa presentation message sa kusum tezet binong to buka gu semnong labdo agya toxic future. So sorong sari sa presentation re pumtong plong kang munong ka kan ligada gu ari presentation re migdum aringka thunong atsu. So well, it is a dream comes true for all the scientists and the researchers. In the latest trend, I'm saying, once they have a patent. So this is our dreams comes true for all over the world, across the world, the scientists and researchers. So in my case also, my dream was to come up with a kind of patent and support the nation. So this dream came true. And you can see here, so yeah, this is the patent on 28th May 2025. I got my technology being patented. It is a huge not only for the Lepcha community but for the nation and the state of Sikkim. So in a brief, I want to explain something about, in a brief, just brief, what exactly the patent is. So patent plays a crucial role. I don't know what's happening. Sorry for inconvenience. Please show me the first one. Escape and show me the first one. Sorry for the inconvenience. So we know that patent is nowadays one of the demand of the government also, so that we can compete across the world, globally, in national scenarios also. Yeah, up to this. So a patent, you can see a pretty crucial role in a country's development, incentivizing innovation, fostering economic growth, and promoting technology advancements. Once you come up with this kind of things, US is now leading this scenario, okay? So in 2024, they have dominated these things. That's why they have, actually they have adopted this 100 years ago. But, so they are one of the developed nations, and they are in high positions right now, and followed by Japan in 2024, and of course China. Now, latest scenario, China is now indulged that second position, is coming second position. In case of India, if you see, India is now also coming up with this kind of patents. They have understood the value of patents for the development, for the GDP development, for the financial development, economic growth. Patent has a huge role to play, because we, till now, with what we have been doing is we are adopting the technologies from across the world. For that, we have to pay the things. So once we have patent, then we can go for the commercial, globally also we can advertise our own technology. This is what our nation was lacking. But the good thing is that India is now also coming up with this kind of things. So India, from into 2024, if you see, 3,520 these patents were granted. So among these 3,520, in latest scenario I'm saying this, among this one is mine, that is, my patent is granted from South Africa, International South Africa patent. So my topic was on this method of planting Thistleana maxima for preventing landslide. So this is the certificate I have received. So it is a huge news for us. And so overview of the concept of what exactly is there in my patent. So this is the canopy system of this Thistleana maxima. The planting of this Thistleana maxima will help to mitigate the landslide in the Himalayas of the world. So canopy system shields the landmass from the high intensity rainfalls. We know that landslides are very frequent in case of rainfalls, whenever there is a rainfall. Right now also it is happening, so it is very dangerous. So whenever there is a rainfall, what happens is Thistleana maxima, which we term as Fyongmu in Lepcha, and it is a tiger grass plant, okay? So the canopy system here, you can see the greenish part. So this will help it will shield the landmass from the direct bombardment of this high intensity rainfall droplets. So this way it will be sweeping down all the rainwater droplets. So another thing is, it has a root shear strength to bind and stabilize the landmass. So this is also the fibrous part of this species is playing a vital role to stabilize the landmass and mitigate the landslide. At the same time, so one of the interesting things, value additions to this invention was, the discovery was that rhizospheric anti-pollutant gene was also found on the rhizospheric soil and between the soil and the root systems. So this is now not only for the Himalayas, for all the cities across the world also it is very, very beneficial, because it is playing an anti-pollutant gene this year that is not Cladophyllophorae species. I have found that. So that's why it has again added the value. That's how it has been scrutinized, and it was found that valued, and it was patented. It took nearly 2 years for scrutiny, whole of this scrutiny I was to go through, and finally I got patented this year. It's a very pleasant year for me. And so the module design. So as per the biomechanical studies base I have designed to identify the root shear strength of this species. Nearly 70, 80 species I have gone through. From that, I was supposed to identify one shrub species. That was a tough job. It took nearly about 4 or 5 years. So after that, I go for bioengineering, measurement to the calibrations to the everything, including the plantations of these species. So next thing is genomic research, which is one of the tough field I have found here. It took nearly about 7-8 years to complete this. So I was to go through. Now I have found that this plant is useful. Now I'll have to understand, I have to justify in front of the world that how this plant is surviving in the Himalayas. So I'll have to go by genomic research. So for that I use biotechnology and bioinformatics. So these are the this is the Cladophyllophorae species you can see here, which is present inside the roots of this Thistleana maxima. Amliso in Nepali we term it. So BTEX means benzene, toluene, ethylene, xylene. Maximally this gene used to biodegrade this kind of pollutions. So these are pollutions which happen because of combustions, because of these vehicles, industries and all. So if you go for afforestation, or we can say the bioengineering of this, then once we have this, you can see the rhizospheric architecture has also been promoted here, manifested here, which will give rise to Thistleana maxima's expansion. We know that Amliso used to expand, grow in expansion manner. So then it will give a healthy vegetation and mitigate the landslide. So now talking about the PhD doctorate of philosophy. So I have completed it from so I will tell you, I have completed it from West Bengal State University, Government of West Bengal, and Barasat, Kolkata. So in 2022, I have completed under the supervision of Professor Sanjay Guha Roy, who is one of the eminent scientists, who is well-known scientist of the world also, in India also. So my topic was on study of vegetational diversity and microbial rhizospheric associations in landslide-affected areas of East Sikkim for assessment and identification of possible mitigative conditions. So these are some study zones which I have attributed by means of satellites and all. So these are the places where I have chosen as my fields. V2 landslide, Ganeshtok landslide, Namli landslide. 8 landslides were chosen among them. These are Namchebong is there, Amdogolai, Penlong, and Andheri landslide, Janbari landslide. Some are active, some are dormant. So these two are been chosen. These are the landslide free zones because I'll have to compare it. No? So I choose these two fields. You know there is no scares of landslides at all in case of Mirung and Busuk. It's very good news. And very interesting thing is that when I go there, field visit, it was wholly dominated by Thyslona maxima. So that is one of the main reason also. That's how it really helped me to understand more about the Thyslona maxima. So now talking about the pilot survey section, almost 17 science fields I have taken up to complete my PhDs, including the pilot survey, selection of sites, GIS and remote sensing, quadrant GPS also I have taken, meteorological data collections with the help of Government of India's offices here in Gangtok only, plant vegetation diversity, plant identification I have done. So another one is I have to know the status of the landslide region. So for that I have used this formula, erodibility K factor, which is known as. So it was already been used, introduced by Goldman et al. 1986, Weisserman et al. 1971. So these were some of the things I have to justify. So these are all the analyses were done in our bioinformatics software. Our base tools, R Studio was used to calibrate and exhibit all the graph images, including the box plots, bioengineering and mitigators. At the conclusion house, with the support of Forest Department, I have done this, some of the areas I have chosen. So this way I have completed 17 fields I have chosen. So trees also I have chosen, shrubs also I have done, and herbs also have done. So mostly my focus was on shrub species only. Later on I come up with shrub species only. So these are some of the pictures through which I have done. So this VAM study was one of the major component of my studies. So I was supposed to justify the vesicles and arbuscules presence, which are very important for the nutritionist support supplements to this plant. So these I have done a comparative studies both in rainy seasons and in dry seasons. These are the graphs. So among that, after having genomic research for 5-6 years, I have done this, nearly 7 years, and completed this. And gene sequences I have done, and so all these are the genes which are present in the rhizospheric part of this Thyslona maxima. Uncultured Cladophylophora, these are 4 species I found, and Ulmus glutinous also I found. So these are the national level laboratories I was supposed to take help. So including the Northeast Regional Institute of Science and Technology, Nirizoli, Itanagar. So I used the biotechnology lab there. So Tripura Central University, there I learned how to pick up all the microbial organisms which cannot see with naked eye. I was supposed to pick up from this microscope only. I was to pick and crush their DNA and find out the DNAs, what are present there, the genes. So another one's IBSD's lab also I have used. Dr. Pradeep, he helped me a lot. And another one is my molecular lab in the West Bengal State University. Dr. Guharwai's laboratory I have used. So apart from this, some of the laboratories in Delhi, Bangalore, Chennai also I have used, but mostly I have used these 4 lab. So talking about this primers NL1, this NL4 I have used for this. And after having this gene colony session, I have gone through gene sequencing and gone for the analysis with the help of bioinformatics. Since I am a bioinformatician only, so I do it myself there. So these are the gene bank which I have, World Gene Bank, where I have submitted all the genes. These are the genes I have submitted, and the people are also now working upon these genes. So in World Gene Bank, that is known as National Center for Biotechnological Information. So this is my beautiful university, and we know that education is simply a soul of society as it passes from generation to another. So another major achievement I have found was in the Northeast India Award, National Award I was received in 2023 for notable contribution to digital innovation for sustainable development in Northeast India through bioinformatics, BEP, under the category of learning education, BEP, have significantly impacted the region and exemplify a commitment to progress for technology. So I take this platform to thank Government of Sikkim also, of course to our Honorable Chief Minister Sir, Sri Prem Singh Tamangju, for all the help, financial help, support to the department and to my bioinformatics center, as well as to my dear Minister Saab of our Science and Technology Department, Sri Laydong Lepcha, for all the supports, all the time supports. So I really owe this award to the whole of the people of the Sikkim and to my department, the states and all of you. So undergoing strong competitiveness, I was to go through this, and 2 days, whole 2 days, I was to been scrutinized along with all the state scientists. At last, I got this award, so it was a very good moment for me. So these are some of the crucial publications which we have published. So now our center has also been recognized across the world. From Belgium, I have recently completed one research project on bioinformatics. That was a molecular docking-based in silico assessment to predict drug efficacy for appetite regulation to mitigate anorexia in MDR-TV treatment. Actually, she has done her PhD on cancer, but I insisted her to do this because it's very important for TB. In Sikkim. So MDR-TV, she has completed her internship. She was from Ghent. Ghent is coming under 100 topmost university in the world. So she has completed, Dr. Renu. So these are some of the genomic and proteomics and of course drug designing trainings which happen in our centers. I've been leading these centers in the states or whoever scientists, including the doctors, scientists, they are coming to learn the bioinformatics. So I've been disseminating these facilities. So thank you so much, sir.